In this video, we're going to look at how the cell manages to get 2 meters of DNA packaged into the nucleus. Now, hopefully, through your additional reading, you're familiar with the structure and building blocks of DNA. And so if I start talking to you about double helices, the sugar-phosphate backbone, and the bases adenine, guanine, cytosine, and thymine, you will know what I'm talking about. So we have 2 meters of DNA, which is 2 nanometers or 2 times 10 to the power of minus 9 meters wide in each diploid cell of our body. And we need to pack this into a nucleus that has a diameter of about 6 to 10 micrometers. How is that possible? The human genome consists of 3 billion base pairs, and these bases are not in one big long double helix, but are in fact cut up into 23 chromosomes. That is 22 autosomes and a sex chromosome. And of course, we have 2 versions of each chromosome, the one we got from our mother and the one we got from our father. And so this gives us the final base pair count of 6 billion bases per cell. So the first stage of packaging is to split the 3 billion bases into 23 chromosomes. And these chromosomes can actually be viewed down a microscope. The image you can now see is what is known as a karyotype. And this shows the chromosomes from a person's cells. Normally, if you look down a microscope, you wouldn't be able to see the chromosomes. But if you can capture the cell at a key point in cell division, they are visible, which is what has happened here. In the image, the chromosomes have been stained, which gives them the banding pattern, photographed, and then paired up. And when you look at the image, one thing is immediately apparent. And that is, not all the chromosomes are the same size, and that some are larger than others. That is, some chromosomes contain more DNA than other chromosomes. As you can see, there are 22 chromosome pairs, plus 2 X's, which means this person is a female. If there was an X and a Y chromosome, as shown in the panel, then the person would be a male. Doing such chromosome spreads can be useful for detecting major swaps of genetic material between chromosomes, or the duplication or absence of chromosomes. Such changes can often lead to genetic diseases, so these sorts of images can have a use in a clinical setting. Now, if we look at the chromosomes in the karyotype, you will see that they have a similar structure. The chromosomes have 2 long arms, 2 short arms, and a central region where the arms join. Also, the arms have a series of stripes that give them a distinctive pattern. In this drawing of a chromosome, we can see the common structural elements. We have the central region where the arms join, and this is called the centromere. The ends of the chromosome, which have an important role in preventing the chromosome from being degraded, are called telomeres. And these regions consist of repetitive nucleotide sequences. The telomeres have an important role in cell aging, and they also shorten as the cell divides and ages. The short arm of a chromosome is called the P arm and the long arm the Q arm. You can remember the short arm is the P arm, as the P is for petite, which means small in French. The Q arm is called the Q arm just because it's the next letter after P in the alphabet. As you can see from this drawing of the banding patterns on a chromosome, which is called an ideogram, the different parts of the chromosome have different identifiers. These identifiers or addresses help geneticists to map the location of specific genes and also the location of mutations that may be involved in genetic diseases. For example, this is chromosome 17. We have the short P arm and the long Q arm, and we have regions, bands and subbands. So if we are told a gene is located at 17q12.3, note that you say 1, 2 and not 12, then this tells us the gene is on chromosome 17, the long Q arm region 1, the regions are numbered out from the centromere, the 1 denotes the region, the 2 is the band in the region, and 0.3 is the subband in band 2. So how are the 2 meters of DNA packaged into the nucleus of our cells? Well, as I said, the first step is to put it into chromosomes. However, the string of DNA in the chromosomes is not just left to float around the nucleus and occasionally condense for cell division, it is also further compacted so it takes up less space. The DNA interacts with proteins called histones and also some non-histone chromosomal proteins to form a tightly wound complex that can package the DNA into the limited space of the nucleus. The protein DNA complex is called chromatin. In the first stage of packaging, the DNA interacts with the protein complex called a histone. Each histone is an octamer that consists of 2 of each of the 4 histone proteins H2A, H2B, H3 and H4. The histone subunits are in fact quite small and consist of only 102 to 135 amino acids. The subunits carry a net positive charge and this allows them to interact with the negatively charged DNA. The assembled histone is about 11 nanometers in diameter and 5.5 nanometers high. The DNA then wraps 1.65 times around each complete histone protein, which results in a structure that when seen down an electron microscope is often said to look like beads on a string. The DNA string between each histone DNA bead is about 5 to 60 base pairs long, with an average length of 33 base pairs. The histone DNA complex is called a nucleosome. The histone complex has a circumference of around 35 nanometers, and with 1.65 turns of DNA around the circumference, it means that about 58 nanometers of DNA can be wrapped around the protein. So that means we are already seeing some compression, as 58 nanometers of DNA is now only 5.5 nanometers long. Next, a 5th histone protein, H1, attaches to the nucleosome to form the chromatosome. The chromatosomes then coil and stack on top of each other to form a compact 30 nanometer fibre called chromatin. The chromatin is then looped and packaged using the non-histone chromosome proteins I mentioned earlier to produce an even more compact structure that contains a series of loops and has a diameter of 300 nanometers. The structure is then further compressed to 250 nanometers, which is then coiled to give the 700 nanometer wide structure of the chromosome. The level of packaging and compression we are seeing here is impressive. For example, chromosome 1 in a human is about 249 million base pairs, and if we remember our maths from earlier, each turn of the helix is 10 bases and rises 3.4 nanometers. Hence, this means the DNA in chromosome 1 is about 8.5 centimeters long. However, the condensed chromosome 1 seen during cell division is about a 10,000th of that size. Even during interphase, when the chromosomes are relaxed, there is still a 1,000-fold compression. Basically, thanks to a series of wrapping, coiling and more coiling events, the 2 meters of DNA is squashed down into the smallest possible space to fit inside the nucleus of a cell. So that's it. By the wrapping of DNA around histones and then the coiling and stacking of these DNA protein complex, followed by some further folding, we are able to get the DNA into the nucleus of a cell.